A pleasant evening to all gathered here for the 5 days national workshop on CSIR-UGC-NET-JRF LS preparation in life sciences conducted by Botany for You, an initiative taken by a forum of professors to motivate, encourage and inculcate the art of learning strategies of NET examination online during this COVID-19 pandemic. The objective of this workshop is to make life science students to master the entire syllabus with a positive attitude and to compete with other students all over India and to complete their exam successfully. Dear participants, I feel privileged and honored to be a part of this forum and I wish you all a great success in the examination. I hope you all will enjoy all sessions and learn fruitfully. Today's topic is organization of organelles in cells. Coming to the introduction, cell is the basic unit of life, structural and functional unit of an organism, the smallest unit capable of independent existence and performing the essential functions of life. Anton von Leeuwenhoek was the scientist who discovered cells. Cell theory states that bodies of an organism are made up of cells and their products. Cells are units of both structure and function of living organisms. Viruses are exception to cell theory as they are not composed of cell. They consist of nucleic acids surrounded by a protein shell and are incapable of independent existence, self-regulation and self-reproduction. Cell theory was proposed by three important scientists. They were Matthias Claden, Theodor Schwann and Rudolf Virchow. Matthias Claden stated that all plants are made up of cells. Theodor Schwann stated that all animals are made of cells, and Rudolf Virchow stated that all cells came from pre-existing cells. This is the image of the cell, which shows the plasma membrane that protects a cell, which contains the nucleus, which stores the genetic information. It has ribosomes, which is important for protein synthesis. It has mitochondria, the energy rich or powerhouse of the cell. It contains endoplasmic reticulum, smooth endoplasmic reticulum and rough endoplasmic reticulum. It has centrioles, microvilli and the lysosomes. This is the image which shows the plant cell and the animal cell. Here you can make out the differences between the plant cell and the animal cell. The animal cell has got the cell membrane with centrioles, vacuoles, cilia and the lysosomes, whereas the plant cell, it has the cell wall with the chloroplast, which contains the chlorophyll, which gives green color to the leaves of the plants. It has vacuoles and plasmodesmata. Cell size and shape. Generally, the cell sizes ranges between 0.2 to 20 microunit. The smallest cell is pleuropneumonia-like organism or Mycoplasma gallisepticum. The largest cell is an egg of ostrich, which measures as much as 6 inches in diameter with shell and 3 inches without shell. Great variability in cell's shape might be spherical, polygonal, disc-like, cuboidal, columnar or spindle-like. It is related to the position of the cell. It might be flat on surface cell or polygonal in cortex. Function of the cell. The main important cell might be RBCs or red blood cells, which are biconcave in shape and pass through the capillaries and carries carbon dioxide. There are several types of cells. On the basis of the nature of nucleus, they are classified into two types, prokaryotes and eukaryotes. Prokaryotes, the genetic material is not organized into nucleus and all membrane organelles are absent, so there is no envelope in the system. Prokaryotic cells, in Greek, pro means before and karyon means nucleus. The best examples are bacteria, archaebacterial, mycoplasma, spirochetes, rickettsia, chlamydomonas and cyanobacteria. Eukaryotic cells, in Greek, the term eu refers to true and karyon nucleus, which has a well-defined nucleus with membrane-bound organelles. Examples are higher plants and animals. This shows the difference between prokaryotes and the eukaryotes. A prokaryotic cell, the best example is a bacterial cell which has an envelope that consists of a tightly bound three-layered structure. The outermost glycocalyx followed by the cell wall and then plasma membrane. Bacterial cell wall is made up of peptidoglycan, which is divided into two types based on the nature of the peptidoglycan. Gram-positive bacteria that has many layers of peptidoglycans and teichoic acid, and gram-negative bacteria which has few layers of peptidoglycan surrounded by a second lipid membrane containing lipopolysaccharides and lipoproteins. A special membrane structure is the mesosome, which is formed by the extension of plasma membrane into the cell. It aids cell wall formation, DNA replication, and distribution to the daughter cell, respiration, secretory processes to increase the surface area of the plasma membrane and enzymatic content. They have thin filamentous extension from the cell wall called flagella, pili or longer, thicker tubular outgrowths which develops in response to fertility factor in gram-positive bacteria, so they are called as sex pili. They help in attachment of the recipient cell and forming conjugation tube. Fimbriae are formed in large numbers. They help in the attachment of bacteria to solid surfaces or host tissues. Eukaryotic cell. In eukaryotic cell, there is an extensive compartmentalization of cytoplasm through the presence of membrane-bound organelles. It possesses an organized nucleus with a nuclear envelope. It has a variety of complex locomotor and cytoskeletal structures. All cells are not identical. Plants and animal cells are different as the former possess cell walls, plastids and larger vacuoles which are absent in animal cells. Mitochondria. Mitochondria are filamentous or granular cytoplasmic organelles of all aerobic cells of higher animals and plants and also of some microorganisms. It includes algae, protozoa, fungi. The fluid containing mitochondria is called the matrix. Mitochondria is the first and foremost important subcellular organelle. History of mitochondria. It was discovered by Kolliker in the year 1880. Earlier, when he observed, he found that mitochondria as intracellular structures. The term mitochondria was coined by the scientist Carl Benda in the year 1898, and Leonard Michaelis was a scientist who discovered that Janus green can be used as a supravital stain for mitochondria in 1990. Structure of mitochondria. The mitochondria are bounded by a double-layered membrane envelope which provides good tensile stability and flexibility to them. The outer membrane are 60 to 70 angstrom thick and the two membranes are separated by an inner membrane space with 60 to 70 angstrom unit. The inner membrane of mitochondria increases surface area by giving out involved tubular structures called mitochondrial cristae. The inner membrane of mitochondria have elementary particles, and these particles represent a special ATP synthase and are involved in the process of oxidation and phosphorylation. This is the structure of mitochondria, which represents the outer membrane and the inner membrane and the intermembrane space. You can observe finger-like projections called as the cristae. You can even see the fluid particle filled within the mitochondria, which is called as the matrix. It also contains some amount of ribosomes and the granules. The outer membrane of mitochondria is quite smooth and has many copies of a transport protein called porin, which forms large aqueous channels through the lipid bilayer. This membrane resembles a sieve that is permeable to all molecules of 10,000 Daltons or less, including small proteins. The outer compartment is called as perimitochondrial space or the intermembrane space. The inner compartment, inner chamber or matrix space, which is filled with dense, homogeneous gel-like proteinaceous materials called mitochondrial matrix. The mitochondrial matrix contains lipids, proteins, and circular DNA molecules. Functions of the mitochondria. It performs most important functions such as oxidation, dehydrogenation, oxidative phosphorylation, and respiratory processes. In an animal cell, mitochondria produces nearly 95% of ATP molecules. Coming to the next subcellular organelle, chloroplast. It was coined by Nehemiah Crew and Anton von Leeuwenhoek. Chloroplasts are the organelles present within plants and algae that carry out photosynthesis, a process that utilizes the solar energy and carbon dioxide to synthesize carbohydrates and releases oxygen. They are a type of plastids that contain their own genome and also exhibit maternal inheritance. Structure of chloroplast. They are planoconvex lens-like structures about micrometer in diameter and 2 to 3 micrometer thick. The chloroplast comprises a double-layered envelope, a granular stroma or matrix, a lamellar system, the osmophilic globules called plastoglobuli, and the chloroplast DNA. Plastoglobuli, they are nothing but osmophilic bodies, lipid-rich granules in stroma. This is the structure of the chloroplast, which contains the outer membrane and the inner membrane. Both the membranes are covered by an empty space called as the intermembrane space. You can see the granum, the stroma, and the thylakoid membranes filled with the starch granules and the ribosomes. Chloro means green, plast means living. The pigment chlorophyll imparts green color to the plants. Chloroplast never originates as de novo. They multiply by a fission, a process that implies the growth of daughter organelles. During the development of the chloroplast, the first structure that appears is the so-called protoplastid that later develops into chloroplast. Stroma is nothing but it fills most of the volume of the chloroplast. It constitutes nearly 50% of the protein composition of the chloroplast and contains ribosomes and DNA molecule. Thylakoids, they are nothing but flattened and closed vesicles arranged as a membranous network. It may be stacked in the form of neat coins forming grana. Nearly 40 to 50 grana in the matrix of chloroplast are seen. Thylakoids include light-absorbing pigments, a complex chain of electron carriers, and an ATP synthesizing apparatus. Functions of chloroplast, they play an important role in photosynthesis, a process that comprises of two phases, light phase and dark phase, which utilizes sunlight, carbon dioxide, and ATP molecules to synthesize carbohydrates such as starch. Vacuole is the next subcellular organelle. This is the image of the vacuole. Vacuoles are membrane-bound sacs within the cytoplasm of a cell that functions in several different ways. Many plant cells have a large single central vacuole that typically takes up most of the room in the cell, nearly 80%. The central vacuole in plant cells is enclosed by a membrane termed as the tonoplast, an important and highly integrated component of the plant internal membrane network system. This large vacuole slowly develops as the cell matures by fusion of smaller vacuoles derived from the endoplasmic reticulum and Golgi apparatus. The central vacuole stores salts, minerals, nutrients, proteins, pigments, and helps in plant growth and plays an important structural role of the plant. These are filled with water to the point that they exert a significant pressure called as turgor pressure against the cell wall. This helps maintain the structural integrity of the plant along with support from the cell wall and enables the plant to grow much larger. They play an important role in molecular degradation and storage. Vacuoles often store the pigments that give certain flowers their colors, which aid them in the attraction of bees and other pollinators, but also can release molecules that are poisonous, odoriferous, or unpalatable to various insects and animals, thus discouraging them from consuming the plant. This is the image of the endoplasmic reticulum. You can observe the two types of endoplasmic reticulum, smooth and the rough, with the nuclear pores, with the nuclear envelope, and the central space and the cisternae. Endoplasmic reticulum was discovered by an Italian scientist, Emilio Verratti. In the year 1945, it appeared like a sac-like membrane of the endoplasmic reticulum, which was observed in the cytoplasm of chick embryo cells. It was coined by Porter and Fullman in the year 1952. It is present in almost all eukaryotic cells. These are found to be absent in mature erythrocytes, ova, embryonic cells, and prokaryotes. This ER, it occupies most of the cytoplasm. Endoplasmic reticulum appears to arise from the outer membrane of the nuclear envelope by outfolding or from the plasma membrane by infolding. The smooth ER appears to arise from rough ER by the absence of ribosomes. Thus, two different morphological types of ER are formed, such as rough endoplasmic reticulum RER and smooth endoplasmic reticulum SER. Physical structure of ER, it is a three-dimensional network of intracellular organelle. It is formed by three types of elements such as cisternae, tubules, and vesicles. Cisternae, they are nothing but the flattened, unbranched, sac-like structured element. They lie in stacks parallel to one another. They bear ribosomes on the surface, therefore appear rough. Tubules, they are irregular branching element which forms a network along with other element. They are often free of ribosomes. Vesicles, these are oval and rounded vacuole-like element. These are also devoid of ribosomes. Molecular structure, it is composed of two layers of phospholipid molecules sandwiched by two layers of protein molecules like other membrane in the cell. Protein transport, as proteins are formed in the endoplasmic reticulum, they are transported through the tubules toward proteins of the SER that lies nearest to the Golgi apparatus. At this point, small transport vesicles composed of small envelopes of smooth ER continuously break away and diffuse to the deepest layer of Golgi apparatus. Inside the vesicles, both the synthesized proteins and other product from the ER are present. The Golgi apparatus can be observed using both light and electron microscope. It is also called as Golgi complex. which was discovered by an Italian physician and Nobel laureate Camillo Golgi in the year 1898. Its microscopic structure was discovered by Dalton and Felix in the year 1954. This is the structure of the Golgi apparatus, which shows the cis phase and the trans phase of the Golgi apparatus, its secretory vesicles, the cisternae, and the lumen. Structure of the Golgi complex. It is closely related to the endoplasmic reticulum in both proximity and function. The Golgi complex consists of flattened vesicles known as cisternae, central stack of parallel and many peripheral tubules. It consists of 4 functional distinct regions: cis phase, middle cisternae, trans cisternae, and trans phase. Each region contains different enzymes which selectively modify the contents depending on where they reside. This is the diagram that represents the functions of endoplasmic reticulum. Synthesis. It is also a major site of carbohydrate synthesis that includes the production of glycosaminoglycan. Sulfation, another task that involves the sulfation, which means the addition of sulfur group. Certain molecules pass through its lumen via sulfur transferases that gain their sulfur molecule from a donor. Protein modification starts in the endoplasmic reticulum after translation, and addition of 2N-acetylglucosamine and mannose occurs. In Golgi, glycosyl transferases and glycosylrases are present that helps in the addition and removal of sugar residues in N-linked oligosaccharides. Apoptosis. The Golgi has a putative role in apoptosis, a newly characterized protein, Golgi anti-apoptotic protein, almost exclusively resides in Golgi and protects cells from apoptosis by an undefined mechanism. Phosphorylation. The phosphorylation of molecules requires energy in the form of ATP, and it is imported into the lumen of Golgi, utilized by resident kinases such as casein kinase 1 and casein kinase 2. One molecule that is phosphorylated in the Golgi is apolipoprotein that forms a molecule known as VLDL, which is a constituent of blood serum. Vesicular transport. Vesicles leaving rough endoplasmic reticulum transport to the cis phase of Golgi apparatus and fuse with the membrane and empty the contents into the lumen. Molecules inside the lumen are modified and sorted for transport to the next destination other than endoplasmic reticulum and Golgi apparatus, moves to trans phase and gets placed on either side of the 3 vesicles: exocytosis, secretory, and lysosomal vesicles. Cell specific functions. Formation of cell wall and cell plate in plant tissues, acrosome development in sperm cells, secretion of zymogen in the exocrine cells of pancreas, secretion and transformation of lipid in the liver cells. This is the structure of the lysosome with its lipid bilayer and its membrane, and with the glycosylated membrane transport proteins and the hydrolytic enzyme mixtures. Introduction. Lysosomes are discovered in the early 1950s by Christian D. Dew, who later received a Nobel Prize for this work. The lysosomes are about 0.5 to 1 micrometer in diameter and surrounded by a single membrane. The name lysosome is derived from the Latin word lyso or lion, means to dissolve, to release, or to loosen, and soma means body. These structures are termed the digestive system of the cell, or suicide bags or suicidal sacs. They are present in animal cells, while in yeast and plant cells, same roles are performed by lytic vacuoles. They vary greatly in size and shape. Like secretory proteins, lysosomes are synthesized on rough endoplasmic reticulum, transported to the Golgi complex, and then packaged into the vesicles that can become phagosomes. There are 2 types of lysosomes, primary lysosomes and the secondary lysosomes. Primary lysosomes, they are nothing but small sac-like structures enclosing enzymes synthesized by the rough endoplasmic reticulum. Since they store enzymes, they are also said to be storage granules. Secondary lysosomes are formed by the fusion of the primary lysosome with the phagosomes. They also contain engulfed materials and enzymes. The materials are progressively digested by the enzyme digestive vacuole. This is the entire picture of lysosomes with the process of exocytosis, pinocytosis, and phagocytosis. Lysosomal enzymes have the common property of being acid hydrolases, hydrolytic enzymes with a pH optimum around 5. It includes 5 phosphatases, 14 proteases and peptidases, and 2 nucleases, 6 lipases, 13 glycosylases, and 7 sulfatases. It contains over 50 hydrolytic enzymes that degrades proteins, nucleic acids, carbohydrates, and lipids. Functions of lysosomes. Heterophagy. It is the lysosomal digestion of extracellular materials by the process of endocytosis, which is of two types, phagocytosis and pinocytosis. Phagocytosis is the cellular process of ingestion in which the plasma membrane engulfs substances and pinches off to form a particle containing vacuole. Pinocytosis is the process of engulfment of fluid particles through the plasma membrane. Autolysis, it refers to the digestion of own cells by the lysosomes. It is also known as programmed cell death or apoptotic lysis. It occurs during amphibian metamorphosis, insect metamorphosis, and menstruation. Extracellular digestion. Digestion of materials outside the cell is called extracellular digestion. Lysosomes release enzymes outside the cell by exocytosis and brings out digestion. Extracellular digestion takes place during bone erosion process. Fertilization. During fertilization process, acrosomes of sperm head ruptures and releases enzymes on the surface of the egg. These enzymes digest the egg membrane and provide way for the entry of sperm nucleus into egg. This action also activates the egg for the developmental process. Motor proteins. They convert chemical energy into mechanical energy that is used to generate force or move the types of cargo that includes vesicles, chromosomes, mitochondria, and proteins. They can be classified into three types, mainly kinesins and dynesins that move along the microtubules and myosin that move along microfilaments. The binding of ATP and its hydrolysis provides energy for them to travel. Microtubule organizing center. Centrosome. In an animal cell, the centrosome has a pair of centrioles, each with triplets of microtubules arranged in a ring. The centrioles are surrounded by an electron-rich pericentriolar matrix. Microtubules are the central structural supports in cilia and flagella. Both can move in unicellular and small multicellular organisms by propelling water past the organism. Microfilaments. The thinnest class of the cytoskeletal fibers are solid rods of the globular protein actin. An actin microfilament consists of a twisted double chain of actin subunits. Microfilaments are designed to resist tension. With other proteins, they form three-dimensional network just inside the plasma membrane. In muscle cell, thousands of actin filaments are arranged parallel to one another. Thicker filaments composed of motor protein, myosin, integrate with the thinner actin fibers. Myosin molecules walk along the actin filament, pulling stacks of actin fibers together and shortening the cell. In other cells, these actin myosin aggregates are less organized but still cause localized contraction. In plant cells and other, actin myosin interactions and sol-gel transformation drive cytoplasmic streaming. This creates a circular flow of cytoplasm in the cell. This speeds the distribution of materials within the cell. I conclude with the words of Mr. Jim Rohn: formal education will make you a living, self-education will make you a fortune. All the best, dear participants. I wish you success in your future endeavors. Thank you.